Yeah, thank you for that introduction. As Sarah said, I will briefly be going over some of the highlights and key conclusions from that review that I wrote last year, and that was with Dr. Alicia Quant. And my email is here. If there's any questions that we don't get to by the end of this, or if anybody thinks of something later, feel free to reach out my way, and I'll do my best to help you answer whatever questions you may have. Um, so just to start, it's a little bit different. Normally, when I talk about diatoms, the people I talk to them about don't know much about them. So I go through the whole what's the frustule? How do we know things about them? How do we identify them? Today, I'm going to skip that portion, but I will just briefly mention why diatoms are important. And this is just something I want everybody to keep in the back of their head as the talk continues. So, just to start, they're responsible for somewhere between 1/4 and 1/2 of global oxygen production. I'm sure all of you have heard the slogans like every other breath or thank a diatom today. And largely, this refers to a lot of the air we breathe does come from diatoms. In addition to that, they're ubiquitous, so found pretty much everywhere. This includes almost all bodies of water, the ice on top of mountains, the Arctic, the Antarctic. They're in terrestrial things like soils. And then, as Sarah said, my start with diatoms was studying Orthocera, and specifically it was Orthocera in waterfalls. So I got to study diatoms from this really cool habitat. They are basal members of aquatic food chains. So really, they're a key component of the trophic web, and really they are one of the bottom members that contributes to food for everything above them. A key component of the silica cycle, as well as carbon captures, especially in oceans. And then more recently, diatoms have been looked at for a variety of human uses. This includes things like biofuels, food, and then they're indicator species for water quality and plenty of other things that we as people use them for and have a lot of use. Shifting gears a little, I will introduce a chytrid, and I'll spend a little bit more time on this, just because when I started, I wasn't familiar with them, and they are an interesting group with some interesting things. So, broadly, what are chytrids? They are a fungi, and you'll see that fungi is in quotes. This is referring to the fact that, historically speaking, the field of mycology, which studies fungus, covered a lot of things. Things today we wouldn't call a true fungus were included in the field. So the term fungi can be thought of similar to algae, where it's kind of a broad catch-all word that includes things that might not be all that closely related. But we can broadly break the fungi down into two parts. The first part is the kingdom fungi, circled here in blue. This could also be considered the true fungi, and then the other half is the protostan fungi, circled here in purple, and this includes the oomycetes and the slime molds. Chytrids fall into the kingdom fungi or the true fungi. And I'm only going to mention this here, but I do think it's something important to keep in mind, especially if anybody goes back into the literature or is looking to study these more. But for a long time, chytrids, which are here in this true fungi, were thought to be closely related to the oomycetes up here in purple. And so if you go back in time, you'll see a lot of work that refers to them together. And you'll often see them referred to as either the phycomycetes or the aquatic phycomycetes, or members of these groups. And there is a book from 1960 by Dr. Frederick Sparrow titled The Phycomycetes, which covers entirely chytrids and oomycetes. But for the rest of today, we'll only be focusing on the chytrids. So, as I said, they are in the kingdom fungi or the true fungi. And here is a fairly recent tree of the kingdom fungi. Um, Thinking of the kingdom fungi, it has what you would consider, or probably the first things that would come to mind, and that's specifically the mushroom forming groups, Ascomycota and Basidiomycota at the bottom. What we consider chytrids, though, is circled here in blue. And again, you'll see that chytrids is in these quotations, as it is also somewhat of a broad catch-all term. At a minimum, though, chytrids does include the groups Chytridiomycota, Blastocladiomycota, and then the Neocalamastagales. So it might seem a little odd that within the broad chytrids, there is specific Chytridiomycota, which in my mind at least, I first thought, oh, those are the chytrids. But no, we include all three of these groups, and there is still some uncertainty in how they specifically orient, but we know that they're at least somewhat closely related. To get into a little bit more specifics about what the chytrid itself is, in terms of habitat, they can be aquatic or terrestrial. Probably the most famous chytrid is Batrachochytrium, which is a terrestrial chytrid, and it's a major parasite of amphibians and is a driver of amphibian decline. Outside their habitat, and just some regards to how they survive, they do form resting spores, specifically to survive when conditions are harsh. Usually, they're unicellular. Not always the case. Some of them do form a cenocytic mycelium, and cenocytic referring to it's a mycelial network that doesn't really have any hyphae or discrete parts. It's kind of just a amorphous blob of mycelial tissue. For reproduction, chytrids do not form sporocarps, but they do reproduce, at least in their asexual phase, via zoospores produced from a special structure called a zoosporangium. There is a more complicated sexual reproductive stage for chytrids. I'm not going to go into it. I don't know a ton about it, but I just wanted to point out that it does exist. Going back to the zoospores, which were produced in this zoosporangium, they are a lot of the reproductive characters. They do have a smooth posterior flagellum, and this is actually the portion of the life stage that unites the kingdom fungi to the opisthecons. And then specifically, you can think of a chytrid zoospore looks a lot like a human sperm in terms of just how they operate in the environment. And then the last thing that I want everybody to have a notion of just broadly what chytrids are, they do, instead of a mycelial network, typically they employ a rhizoidal network. And so the rhizoidal network is this root-like structure coming off of the thallus of this chytrid. So at this point, we have an idea of what chytrids broadly are, where they fall in the tree of life, and then generally some of the characteristic or diagnostic features of them in their life cycle. But what do they actually do? Some are saprobes, some are also parasites, and then additionally, some are both. So at times, one might be a saprobe if there's a lot of dead and decaying matter that it can utilize. If that dead and decaying matter goes away and a host that it can parasitize comes along, it might switch strategies and vice versa. So really, some of them can be quite opportunistic. Specifically to diatoms, what they do though, we do know that some are known to be host specific. Taking a quick step back, I will say when I'm referring to diatoms from here on out, I'll only be talking about the parasitic ones. I do believe there are some chytrids that are saprobic specifically to diatoms, but I didn't focus on it for the review as it focused mostly on parasites. So just know there are likely saprobic chytrids of diatoms, but for now we'll just be talking about the parasitic ones. But of these parasitic ones, we do know that they can be host specific. And of these ones, they can have huge impacts on diatom populations. Specifically, they can cause epidemics, and these epidemics have been reported to have greater than 90% fatality rates. I think up to 95% fatality rates is at the high end. Typically, when we do see these big epidemics with these big fatality rates, it occurs in early bloom forming species of diatom. And then I will quickly just go through an example from Lake Marseline to kind of introduce this epidemic a little bit more and some of the things we know about it. So for the Lake Marseline example, this was a study published in 1983 by Dr. Van Donk and Dr. Ringelberg. Well, it was published in 1983. The actual study ran from 1978 to 1980. And it was in Lake Marseline, which is a small oligomesotrophic man-made lake in the Netherlands. On the left is just a Google Maps image of the lake, and on the right is a drawn depiction of it. You can see from its shape the man-made nature. In the study, they sampled water at fairly quick intervals from the lake, and they found that the diatoms Asterianella formosa, Stephanodiscus astrea, and Cyclotella compta all had fungal parasites associated with them. Of these three diatoms, only the Asterianella formosa population had a parasite with a high infection rate, and it was specifically that Asterianella formosa was heavily parasitized, infected by the chytrid Zygorhizidium planktonicum. With this information in hand, they looked at a couple things. The one I'm going to focus on for now is temperature. I'll talk about some of the other portions later on, but just for now, only discussing temperature. And that's specifically temperature in regards to the early portion of the year. So think late winter, early spring, when the water is still cold and the air is still cold, but starting to warm, and a new season might be starting. So they referred to temperature in two broad categories. One would be cold years and the other would be warm years. Cold years was in 1978 and 1980, I believe, and a warm year was in 1979. In these cold years, so the 1978 and 1980, this is when the air and water temperature stayed low, or at least took longer to warm up. It's likely that maybe there was a thin ice cover or just other things about the lake that the temperature was not super high. As you can see from the diagram, temperature is the blue bar. Uh, when it was a cold year, the Asterianella formosa populations were able to grow, bloom, and eventually form quite a large population. But the parasite, Zygorrhizidium planktonicum, didn't grow very well. It stayed low, and there wasn't really much of an epidemic seen at all. So we didn't see that huge fatality rate. In contrast, in warm years, so you can see now that blue temperature bar is at the top, the Asterianella formosa population, again, able to grow, it bloomed, had a big population, but its parasite, the Chytrids Zygorrhizidium planktonicum, also had a big boom in population. And eventually, there were so many of the Chytrid, and they were affecting so many of the diatoms that the diatom population did collapse, followed shortly by a collapse in the Chytrid population, as it no longer had a host to utilize for its life cycle. Um, in addition, though, to just these two crashes, in years with the crash, we saw one other thing, and that is specifically that other diatoms, namely for this study, it was species from Fragillaria and Stephanodiscus, greatly increased in size as compared to those cold years. So in cold years, we didn't really see other diatoms form these big blooms, whereas in warm years, once the Asterianella population collapsed, some other diatoms were able to come in and exploit the niches that were opened up. A similar result was seen in a study in 1949 by Dr. Cantor and Dr. Lund in Esthwaite water. And in that scenario, again, an Asterianella formosa was parasitized by a chytrid, had the same population collapse. And then after that population collapse, different diatoms, in that case, it was Fragillaria and Tabellaria species, were able to move in and exploit those open niches. And this just broadly suggests that chytrids play some role in early seasonal succession of diatoms in these bodies of water. To summarize where we at this point, so we know that diatoms are important. We also broadly know what chytrids are. And then we know that when diatoms and chytrids are together, there can be huge impacts specifically on the diatom individuals, the diatom populations, and then whole community of diatom in general. And these impacts are generally referring to these big infection rates with these epidemics of greater than 90% fatality, and then that how these epidemics and interactions may play some role in diatom seasonal succession. But there's still some information we're lacking, namely, not much is known about infection strategies, so specifically how a chytrid infects a diatom. We don't really know if they're highly host specific or if the chytrids are generalists. And then we don't know a whole lot about how diatoms respond to infections, but there are some ideas. To jump into the first one, which is that method of infection. So here's a flowchart that I made and did publish in the review. The flowchart was created with biorender.com, which was pretty easy to use. I will pull apart each part of this diagram, but I wanted to show it in full before doing so. To dive into it, we'll start with A, and this is just broadly, no matter what infection strategy that may occur, the diatoms and the chytrids first have to be in some body of water together. They may not be interacting yet, but they have to be somewhere they can potentially interact. Eventually, for infection to occur, aside from just being in a body of water together, the chytrid also has to be able to find and locate the diatom it wants to infect. From there, it'll have to insist upon it. But just to find it, we don't know exactly what happens, but there is a suggestion that the chytrid is able to orient to diatoms based on some photosynthate that the diatom releases. And from that, so if you think of the diatom, it's releasing these photosynthates. Near the diatom, there's a heavy concentration of them, and then further away, there's a lighter concentration. as the chytrid swims along, it'll sense this lighter concentration and follow the density to get where there's a higher concentration. And that brings it to the host. Once it is to the host, it has to land on it. And then it has the new fun job of trying to get through the frustule, which can be thought of broadly as a glass suit of armor around the diatom. There are two broad suggested ways this happens. They are similar, but there's one key component that's different. So I will start with C. Within C, there are also two possible ways this occurs. The first is that after landing, the chytrid develops this germ tube, this tail-like structure, which then extends and probes along the surface of the diatom frustule until it reaches the girdle region. At the girdle region, it then looks for a gap between two girdle bands. When it finds a suitable gap, it then wedges this germ tube between that gap in order to gain entry to the inside of the cell. The other option for C is similar. Again, the chytrid lands and then it'll develop this germ tube, but instead of the germ tube searching along for the girdle elements of the diatom, it'll instead look for any suitable ornamentation on the frustule that it can exploit. An ornamentation that's been suggested is the raphe and diatoms, and then larger areolae or paralae. Using this ornamentation, the chytrid uses its germ tube to go through the hole in the frustule and gain access to the diatom interior. So for both options in C, the chytrid exploits some frustule weak point. Switching back to D, which is a little bit different, again, the chytrid has to land. From there, it'll develop this germ tube in the same way, but instead of the germ tube searching for a potential weak point in the diatom frustule, it'll actually find any suitable open area on the frustule, and by open I mean just a portion of the silica doesn't have anything going. Once there, it then creates its own opening to get inside. The first suggested method is that the germ tube actually drills a hole through the frustule, and then there's another suggested method that instead of drilling a hole mechanically, the germ tube releases some compound that dissolves the silica of the frustule. So in this one, instead of exploiting a weak point, the chytrid is creating a weak point. I'm happy to discuss either of these more, either at the end if anybody has any questions on them, or via email. I tend to lean towards C. It just seems a little bit more reasonable for me to digest, but both options could be possible. At this stage, we can't really say if one is or one isn't. It's also possible that neither of these are occurring. There's just really limited information studying diatom or the infection process of a chytrid on a diatom. So there's still a lot to learn. But regardless of how the chytrid does get in, once it's in, we do know that it'll develop this rhizoidal network. So this is the root-like structure coming off of, at this stage, what is now a zoosporangium, which has been created. The rhizoidal network steals and takes all of the organic content from inside the diatom that it wants. And then within this zoosporangium, it's producing these new zoospores, which then, once a certain density is reached or all of the organic content has been taken, the zoosporangium bursts and it releases all these zoospores into the water, which can then go and infect new diatoms. So this seems like a pretty sweet gig for the chytrid. Not as nice for the diatom. What does the diatom do in response? There are a number of potential mechanisms and how the diatom may respond. Here they are briefly, and now I will quickly try to go through each one of them. So the first potential response mechanism or defense mechanism is just broadly the red queen hypothesis. And the red queen hypothesis suggests that diatoms evolved sexual reproduction as a whole in order to expand their genetic diversity in response to pressures from these and other parasites like them. This was definitely the most reported defense or response mechanism of the five I'll talk about that I found while I was reviewing the literature. And then just specific to chytrids, which this was talked about a little bit in the papers I read, is there was an example of a study that had two species of chytrid from the genus Zygoflictus. For our purpose, we'll call them Zygoflictus A and Zygoflictus B. Both of these chytrids could infect Asterianella formosa. Specifically, though, they could infect specific strains of Asterianella formosa. The two strains of Asterianella formosa were morphologically identical, but they had some small molecular or genetic difference or came from some different series of generations that separated them. So Zygoflictus A could infect Asterianella formosa strain A, and Zygoflictus B could infect Asterianella formosa strain B. However, when these were crossed, so now you have Zygoflictus B trying to infect Asterianella formosa strain A, and Zygoflictus A trying to infect Asterianella formosa strain B, no infection occurred. Whereas if we go back a step, and when they were matched to their original one, infection would occur, followed by a large epidemic with a big fatality rate. And then when they're crossed, no infection, no fatalities. The other, or the second response is just broadly a hypersensitivity response, and hypersensitivity does refer to a programmed cell death. We go back to the diagram depicting a Chytrids infective cycle on a diatom. In a hypersensitivity response, the Chytrid would be able to go through A, go through B, and then when it gets to panels C and D, it would land on the cell, start its infection, and then the diatom host would just die. And since the diatom host has died, the Chytrid is unable to subsequently go to stage E, where it would exploit all of those resources within the diatom, and it's unable then to develop that zoosporangium and release the zoospores to infect new cells. So this stage never happens. A hypersensitivity response is a altruistic response, so it's not a very good deal for the individual host diatom, but it is a good deal for the population of diatoms as a whole. The third potential response is that somehow diatoms are producing a fungicide. This refers to a study from Ponert 2000. And in that study, it was indicated that mechanically wounded diatoms produced a fatty acid derived metabolite. These fatty acid derived metabolites released unsaturated aldehydes, which then could act as a fungicide. This response is not specific to Chytrids, but it is a potential avenue that could be interesting to further explore and try and document how diatoms refer or respond to infection broadly. The fourth defense mechanism I'll talk about, or response mechanism, is called a deep water seed bank. And this refers to a scenario where some diatoms sink to the sediment, or at least sink out of the active infective zone. And this puts them below the range of survivability of Chytrids. And when they're below that range of survivability, they can then dodge the infection and epidemic. When I say below the range of survivability, I'll talk about this more shortly, but there is some work that indicates diatoms can survive both at colder temperatures and at lower light intensities than their Chytrid parasites. So just to get into this a little more, if you think of a water column, you have your photic zone, and then you have either the sediment or the aphotic zone beneath it. In the photic zone, we have a bunch of diatoms growing, going through their life cycle. As that diatom population gets bigger, there's a subsequent Chytrid population that's going along and doing its life cycle. We'll see the diatom population grows. Simultaneously, the Chytrid population starts to grow. Eventually, you'll have a lot of diatoms and a lot of Chytrids. Well, here, some of these Chytrids will be infecting these diatoms. As the infection goes, especially if it's one of these large epidemic forming Chytrids, the diatoms will start to die. All the while this is going on, some of the diatoms may be sinking just due to an inability to stay suspended in that water column, either to the sediment or out of the aphotic zone. So they're sinking. This epidemic is occurring above them. Eventually, all the diatoms above them will die, or at least most of the diatoms will die. This will be followed by all the Chytrids dying as they no longer have a host. But beneath all of this, we still have these diatoms that sank, able to survive and kind of dodge everything going on above them. Then, as the year continues, the temperature might cool, the water might lose its stratification, and some of these diatoms that had sunk out of the infective range will then be pushed back up into this photic zone. There, they can then start their life cycle, so the diatom will start growing again, and then eventually the Chytrid will come along again and repeat the whole cycle. And so this is part of that seasonal succession thing where year after year, some of these diatoms may have survived beneath the infection and everything above them died. Water turns over, they get elevated back up in the water column, and then the cycle starts over. The last defense or response I'll talk about just briefly is an immune response. I'm only going to briefly touch on this as it was mentioned just once, and I'm not an expert in immune responses, either for diatoms or immune responses in general, but I thought it was worth mentioning in case anyone else finds it interesting, because I definitely found it interesting. And then if anybody's curious to study it more. So the study that does mention this is Friedman, 1952, and it was in this study, it was reported that a diatom survived an epidemic. So a diatom had a chytrid on it. That chytrid subsequently released from the diatom or didn't finish its life cycle for some reason. Um, that surviving diatom was then taken and exposed to a water with a whole lot of the chytrid that couldn't or didn't kill it. And then when it was re-exposed, that chytrid never landed, never re-encysted, and never infected that diatom that had survived. So this just suggests that somehow, if a diatom survived an epidemic, it might not be able to be infected any longer. The details of this are a little sparse, and I did not see anybody else that either replicated it or reported anything similar, but it does suggest some interesting possibilities that maybe something about an infection makes it hard for a diatom to get infected again. And I think it's something that could and should be looked into more in the future. So I've talked a little bit about how diatoms respond, also how they're infected. I just want to touch next on how we got to where we are now. So how we figured out the work or figured out the information we currently have. And so that's just broadly the work that has been done so far. The first I'm going to talk about is light scenario or light experiments. And within light experiments, there's really 4 broad scenarios that were looked into. The first of which is just under light conditions. So think of the diatom and the chytrid always exposed to light. In these scenarios, it was found that when the chytrid was in a sample or whatever with the diatom, but not necessarily near the diatom, it would just swim in a free-flowing manner using its flagella. Eventually, it would near a potential host, and then as it neared that potential host, that free-flowing swimming would change to a more erratic motion. Often this erratic motion would get more erratic as the chytrid neared the diatom, and often many chytrid zoospores would surround one individual diatom until eventually the chytrids were able to land on, encyst, and infect that host. In dark conditions, so no light at all, again, the chytrid zoospores are able to display this free-flowing movement, so they're swimming around. But even if they came directly adjacent to a diatom cell, they never displayed that erratic movement, and they did not congregate or eventually land on, encyst, and infect. So only under the light conditions were infections occurring. The next scenario was going from light to dark. So this is when they started with lights on and turned them off. When the lights were on, the same thing happened as in just an all light scenario. The chytrid would swim freely until it neared a diatom. Then it displayed that erratic motion until it eventually landed on and encysted. If they turned the lights off when the chytrid had landed, but had not yet encysted and infected the diatom, so we went from this light condition to this now dark condition, the chytrid would release. So even if it had already touched its host and knew the host was there, once that light was removed, if it hadn't yet actually gone into the frustule, the chytrid would release and swim away. And then the last one is just going from dark to light, and going from dark to light in the dark again, that chytrid could swim freely, but it wouldn't infect anything. Within 30 seconds of turning the lights on, though, it would go back to, we see that erratic motion, especially when they're near the diatom hosts, and eventually we'll see that infection occur. So broadly, these light experiments are the basis for that photosynthase hypothesis, that, which again refers just to that chytrids lock on to some photosynthetic compound that the diatom releases, and that's how they orient in order to find a host. And everything I just discussed is a summary of a series of articles from Dr. Kees Bruning in the early 1990s, as well as some of the work from Drs. Van Donk and Dr. Ringelberg in the 80s and 90s. And then I just want to mention it briefly, but it's also in that work from Dr. Bruning in the 90s. They looked at different light intensity conditions, and it was found that under limited light, so when light intensity was lower, chytrids developed fewer zoospores in a zoosporangium, and it also took them longer to develop those zoospores. Uh, Additionally, at these lower light intensities, the host Asterianella formosa was found to be less susceptible to infection. And then if you hit a minimum light threshold, the Asterianella formosa could still grow, but the chytrid couldn't. And so that's part of that. When cold years, if you think there might be an ice layer on the water, or the sun isn't overhead super long and the light intensity is not super intense, that early season, why the diatom can grow before the chytrid and how it has this broader survivability range. The next one I'll talk about is temperature experiments. And just broadly, this combined observational and some experimental data, and it found that below 4 degrees Celsius, diatoms could still grow, albeit slowly. Chytrids didn't really grow. And then above 4 degrees Celsius, diatoms and chytrids would both grow, and that's when we would start to see those infections and epidemics. This is the other half of the deep water seed bank suggested refuge or defense mechanism. So if you think about as a diatom sinks out of that top layer of water, gets deeper and deeper, the water gets colder. And so the diatom can still grow, albeit slowly, or maybe it's just resting, whereas the chytrid isn't going to grow. And then moving on, the next study looked at turbulence. This refers just to 2 observational studies, which discussed how turbulence and agitation, specifically in adjacent to dams, in fact, impacted the ability of a chytrid to infect a diatom. So as you may have guessed, there are 3 broad categories that were referred to. They were low, medium, and high, or low, intermediate, and high. In the low disturbance scenario, so this is when further away from the dam inlet, water's not very agitated, it's pretty still. They found that diatoms had a harder time growing. This is likely due to increased settling rates. There's not a lot of agitation in the water to push diatoms back up the water column. In contrast, chytrids could grow quite well. They're able to swim around freely, and they didn't have any water pressure kind of dictating where they go. Shifting to the intermediate or medium disturbance group, it was found that both diatom and chytrids could grow. So there was enough agitation likely to keep the diatom suspended, but there wasn't so much agitation that the chytrid's ability to swim was impacted. And then the last group is the high agitation or high disturbance group. And in this scenario, both the diatom and the chytrid were unlikely to grow. For the diatom, it was likely due to so much agitation that colonies couldn't really form, and the diatom was just beat around. And then the chytrid had so much agitation in the water that it wasn't able to swim and get where it needed to on its own. And so, yeah, in this intermediate group, though, they did see an infection and an epidemic occur, but not either at the low or high end. Across all disturbance levels, though, colonial cells were more likely to be infected than just cells on their own. And then the last thing I'll touch on is just grazing. And grazing largely refers to or focuses on what has been termed the mycoloop. And the mycoloop was a big undertaking and description from the work of Dr. Kugami and collaborators starting around 2007. What the mycoloop is broadly just describes that when fungal parasites are present, the growth of Daphnia and other grazers is more likely to occur versus when fungal parasites are not present, that growth doesn't occur very well for Daphnia and other grazers. So referring to the image, you can imagine we have a diatom and there's no fungal parasites around. It gets grazed by something like a Daphnia. The diatom with its frustule is likely to pass through the Daphnia undisturbed and come out the other end still whole and on its own. Eventually, that diatom will die and it'll sink, and all of the organic content within it will be lost. If you add, however, these chytrids or other fungal parasites, now we have a diatom swimming around. Eventually, it gets infected by a chytrid zoospore or another parasite. Those parasites then take the organic content out of the diatom and use it to produce their own offspring. So in this case, it uses the organic content to produce a zoosporangium, which produces all these little zoospores, which are released and then swimming around. These zoospores can then subsequently be fed upon by Daphnia or other grazers. And when they're fed on, this time the Daphnia or other grazers uptake that organic content and it's not just passed through and lost. Um, and so it's been shown that there's a pretty big impact of this microloop on how much organic content is recycled into the water column. It's also been referred to as the fungal shunt or the FZ link. Each description is a little bit different, but it broadly refers to, like the microloop, the presence of a fungal parasite's ability to transfer some of the organic content that would otherwise be lost from sinking diatoms back into the water column and into the trophic levels. So there are some current limitations that I want to discuss before ending. The first of which is for all of those studies I just talked about, and I'll talk about this more in a minute, Asterianella formosa was the host. So it's a lot of the information we have is just on Asterianella formosa, and then it's very limited in the number of, for those studies I just talked about, the number of actual things that are reported. For each one of them, likely single digit studies, and some of them, like the agitation, as low as 2. But the other big current limitation I want to talk about is that it's likely the diversity of Chytrids parasitizing diatoms may be underreported. Specifically, if we go broad, there's an estimated 1.5 million fungal species. That's the low estimate. The high estimate is as many as 3 million fungal species total. We've described about 150,000 at this point, which, if we're on the low end, leaves 1.35 million undescribed, and on the high end, almost 2.9 million undescribed. So we've described anywhere from 5 to 10% of total fungal species. And then just of these 150,000, only 1,250 of them are chytrids, about 1,250. So it's an area that hasn't been focused on a lot, especially if you think about how big the oceans are, how many bodies of water there are. There's likely a lot of chytrids out there that just haven't been described because not a ton of people look at them. Within the chytrids, about 700 of them are parasitic, and then the other 550 are non-parasitic. Switching to diatoms, there's an estimated 200,000 total potential species. This is an estimate from AlgaeBase, and then about 75,000 have been described so far. That's as of Koselleck et al. 2018, so a couple of years later, likely a little bit more, but really right around 40% of potentially all diatom species have been described, and 60% remain to be described. And then just for all of these, so we have 75,000 diatoms. This should say 150,000 described fungi and not 1.5 million. I apologize for that. 700 of which are parasitic chytrids. We only know of 162 interactions between the two. So this is 162 interactions where there is a diatom infected by a chytrid. And for this, it does include 74 species of diatom and 63 species of these parasitic chytrids. So then just taking that other step back again, it's just likely that there's a lot of undescribed diatoms that could be infected. There's probably described diatoms that could be infected, and there's a lot of undescribed chytrids out there that likely do infect diatoms. So really just a push needs to be made, or a lot of work likely needs to be done to describe more of these and define more of these interactions. And just broadly, we do have a lot of undescribed fungi, a lot of undescribed diatoms, and limited study of these two things together. The other big limitation I want to talk about is that a lot of the information we have is tied to just a few hosts and parasites. So as I mentioned, there's 162 reported interactions total. As you can see, based on the histogram, about 110 of them, 110 of these 162 interactions are only reported once. And then it quickly tapers. Once you get to 5, it's almost non-existent. But you may have noticed, the eagle-eyed viewers of you out there, that there are some of these ones on the right, shown by the arrows, that are reported a whole lot. So at 24, 20 times, 14 times, and 11 times. However, all of these are on the same host, Asterianella formosa. So while some things have been described, reported, and looked at more, the chytrid changes, but the host doesn't. It's pretty much always Asterianella formosa that's being looked at. And again, going back to those studies I talked about, the light, temperature, turbulence, and grazing, the main host focused on was Asterianella formosa. So at this stage, we do know some things, but we can't really extrapolate it to all diatoms. It's more likely that we can extrapolate it just to, this is how Asterianella formosa responds. And so more work will need to be done there to see if it's true for all diatoms, or maybe there's other things that other groups do. And then another limitation is that most of our information is tied just to freshwater systems. So of the 162 interactions, 133 of them are from freshwater systems. There's another one that may also be freshwater. I wasn't entirely sure, so it has a question mark. And then 9 are from brackish habitats and 8 are from marine. And so if you think of how big the oceans are, to only have 8 total known interactions in that whole system, there's probably a lot more going on. And then the last limitation I will just briefly mention here, as it's an interesting point, is that almost all of the information we have is for planktonic diatoms and chytrids. We don't really know anything about the benthos. Nobody has looked at it specifically. For the review, I looked at over 250, somewhere between 250 and 300 references that talked about this, and maybe 2 of them total even mentioned the word benthos. So it really just hasn't been looked at for chytrid parasites of diatom. And then just to start wrapping things up, I've alluded to some of these, but to summarize, I do think there is quite a bit of future work that can be done outside of just what I'm going to say, but these are some of the places I think it would be interesting to start. The first of which is just integrating studies with new diatom hosts and new diatom parasites. As I said, a lot of what we know is based on Asterianella formosa. Looking into new diatom hosts to see if what we found for Asterianella formosa is true for other hosts would be interesting. And then looking at more parasites, both the ones that we know of and potentially ones that are to be described in the future. And then I think a focus on marine diatoms and chytrids would be helpful. Some of this work is being done, but it largely at this stage focuses on broad like monitoring and not the identification of these specific interactions. And then just broadly, even outside of marine settings, just push to identify more interactions as a whole, so that we can get a better sense of what is going on. As again, right now, a lot of what we know is limited to a very, very narrow set of, at this stage, maybe model hosts, and not a broad group of diatoms. And then I just have a brief resources page here, as I do know this gets published in the future. So if anybody does want to get started, these are some places that I looked and I think would be helpful to look at in the future. For some starting points. And then you can also look into the review I wrote, as all of these references are included there. Does anybody have any questions?
Hey, thanks, Gus. We have a smallish group here, so if anyone has questions, feel free to raise your hand and we'll call on you. Otherwise, if you're feeling shy, go ahead and post your questions in the chat and we can ask them there. We've already got questions. Matt, why don't you go ahead and turn on your camera and your mic and ask your question.
Hi, Gus. First, thanks for a really interesting presentation. So as someone who isolates a lot of marine benthic diatoms, I'm wondering what the status of molecular markers for some of these chytrids are. Is there a good reference database of small subunit ribosomal reference markers?
I don't know off the top of my head. I didn't look into it a ton. What I can tell you is there's a number of whole environmental metagenomic data sets that exist. This is where a lot of that monitoring work has gone. And from that, at least they're able to identify however many, whatever proportion of the reeds are fungal. And then within the fungal reeds, they can say if they're from something like Chytridiomycota or not. But I don't know specifically if there's any good markers at that level.
Good, because I mean, I do find fungal reeds within the cultures.
Mm-hmm.
Dozens of them, and I just sort of put those aside. But
Mm-hmm.
if there's a way to identify those as chytrids, then those are potential reports for y'all.
Mm-hmm.
But again, if that's a way to identify them.
I've seen that looked at. The other difficult portion of that is tying it to necessarily what the diatom host might be. So just to read, if you don't specifically see it infecting, you might only see that the diatom host exists in that population with a fungal parasite. So I think that's been a limiting factor in some of that work is just how you know or tie things together.
Okay. Thank
All
you.
right. Thanks, Matt. Hey, Pat, go ahead and turn on your camera and mic and ask questions.
Hey, Gus, thanks for a great talk. Tell me, please, so you're saying for future work, we should look to marine environments. And so how many of, if you were going to break down this group, these chytrids, into freshwater and marine, not from their descriptions, what's the proportion of these things that have been described that would be freshwater, and how many of them are from marine environments?
I don't have a number off the top of my head. There is that about 700 known parasitic ones. Almost all of the descriptions I do believe are for freshwater, though. Most of it was described somewhere between the 1930s and 1980s. And it was a lot of the work of someone like Dr. Hilda Cantor. And that was in freshwater systems and somewhere in the UK.
Yeah, and Sparrow's work is all freshwater. So I wonder if maybe the group is just not well represented in marine systems, rather than although Matt just gave us some hope that there's maybe more there than perhaps we perceive from morphology.
Yeah, going back to what Matt said too, there are these environmental metagenomic data sets which do suggest there are big or at least large populations of these chytrid fungal parasites in marine environments. They just haven't been looked into very specifically.
Thank you.
Yeah. Hey, Gus, while we wait for others to ask questions, let me just, one of the things you really highlighted was the specificity of the chytrid species to specific diatoms. Is there any evidence of more generalist chytrids that are capable of infecting diatoms in general?
There is some. Again, I don't remember what the chytrid species is off the top of my head, but it's one that we're looking into a little bit more now, and it has been shown to infect just diatoms broadly across the whole diatom tree of life. So something like the radial centrics can be infected by something that also infects something down in the raphids.
Oh
so
my.
at least some of them have this broad impact, but hasn't been looked into a ton. So there's probably only a few right now that are known. I don't know if any have actually been reported to infect something like the multipolar centrics, but if it can cross the whole tree, it's probably likely that the multipolar centrics are too.
Interesting. Michaela just made a comment into the chat that said she's nervous about modeling diatom ecological responses without knowing much about the fungal infections in these data sets. Any just sort of thoughts on how to reconcile that?
It's a tough thing. I really didn't know anything about chytrids when I started. There's still a lot I don't know about chytrids. I really know a narrow set of things about them in regards to diatoms. My recommendation would be to find maybe a mycologist to work with. I think on the same end, there's mycologists that look at chytrids and they don't talk to phycologists. And so there's things that if there was some interaction going on, both parties would probably benefit.
Cool. Sarah, go ahead and ask your question, please. Uh-oh, Sarah froze up. Hey, Tom, why don't you go ahead and ask your question?
Okay, hi, Gus. Thank you for the introduction to chytrids. Or chytrids, however we may pronounce them. I see them quite often in Catastrophes and some of the marine stuff. But I had a couple questions and a comment for you. So, do these chytrids affect or infect other algal groups, other algal classes?
Yeah, they've been shown to infect, I think, pretty much anything that you can find in a body of water. And then some of them are also, I think the term is hyperparasite. Somebody correct me if I'm wrong, but there are chytrids that will infect chytrids that are infecting something else too.
Okay, and I just wanted to mention this. I don't know if you're familiar with this. I'll hold this up if I could. I have a book by Drebus, And there's a whole bunch of pictures of fungal parasites at the very end of that book. Um, just just kind of interesting. And a final question. You mentioned one possible mechanism you're looking into is that they may create their own entry into the cell. So is there any kind of evidence with SEM looking for a scar, somewhere, a hole or something like that that may be produced by these chytrids, or if not in diatoms, in other algal groups?
Not that I know of. It's something that I want to look into. I believe there's a potential way that you can look through an SEM with the chytrid and the diatom both being present. I need to look into it more, but I think that'd be interesting so that we could see at this really fine resolution what's actually happening. Because again, a lot of this is somewhat dated, and there isn't a huge set of information to go off of. It's pretty limited in terms of what has been done.
Sure,
really,
I have a problem that down on my microscope where I get a fungus that colonizes one of the filters on the camera, and it actually etches it. So
Oh yeah.
I know these guys can go through glass coating. So yeah.
Yeah.
Again, thank you. This is a great talk.
Yeah, thank you. I think your email a while back was part of what spurred this to happen too.
Cool.
Awesome.
Thanks, Tom. Got a couple questions in the chat. Karen asked, are there existing ongoing diatom chytrid systems that are already isolated in the lab and can be used for experiments?
The broad monitoring I referred to is more of these like whole environmental samples. Are there some work from Dr. Bettina Scholes out of Iceland? That might be a good place to start to answer that question. In terms of existing ongoing diatom chytrid systems that are already isolated, there is something, it's called, the acronym is cesium. I believe it stands for the collection of zoosporic U fungi at the University of Michigan. It's a new collection of only chytrids that's maintained by the University of Michigan. And they have a whole bunch that you can order and they'll send them to you. I've ordered one so far and it's been really successful at staying alive in culture. I've had different issues keeping or growing some diatoms I'm hoping to look into more. But eventually our hope is to do something, I think, like what you're saying, to get this laboratory system going.
Cool.
So that we can study these experiments in a controlled setting.
Are the chytrid fungi grown? Can they be grown on plates, or do you have to provide them with a dead or a diatom to feed on?
At least the one I ordered, which is one, if you look in the review, there's a big table that records every published interaction between a diatom and a chytrid. So where we have a chytrid that we know parasitizes a host diatom, they had one in the collection. Out of that, I believe it's Rhizophidium globosum, and it can apparently be grown on a media or in a broth. So you can grow it away from the diatom, and then once you have both your diatom population and your chytrid population, you can then experimentally infect them.
Very
so
cool.
I think it refers to how some of them have this, they can exploit or employ multiple life strategies, just kind of based on what's the most opportunistic.
Very cool. Sarah, you've got your hand up and you wanna ask your question and then close us out, please?
Sure, yeah, I had an unstable connection. I dropped off earlier. Gus, thanks for just a really fascinating topic. And beyond the molecular markers, are there any particular stains that are useful for chytrids?
Yeah, that's how we know. The portion that we know pretty well is once the diatom's in, we can see that rhizoidal network really easily. I believe it's calcoflower white or maybe blue. There's either a white or a blue stain, and that you can add it. And then if you have a population of infected diatoms, all the ones that are infected, you'll see this rhizoidal network in either white or blue, pretty easily discernible.
Have you tried that stain? Is it
I haven't
super?
yet.
Carcinogenic.
I don't know. It might be. I know some of those things are. I haven't tried it specifically yet. We're still at the getting the cultures of each thing established before infecting them in a controlled setting. So eventually, the hope is that we will have that to look at. Yeah.
Yeah, great. Yeah, thanks so much.
Well, we reached the top of the hour. I want to thank Gus for a really interesting presentation. I wrote 2 pages of notes while you were talking, Gus. And I do want to again remind everyone that on April 9th, Tuesday, April 9th, we'll be having our next Diatom Web Academy. I'll have a little bit of a twist towards some cyanobacterial dynamics, but I'm sure there'll be some diatoms as part of the picture. So thanks everyone for joining us today. Great to see everyone, and hope everyone has a great rest of your week.
Bye. Thank you.
Thanks, everyone. Take care.
Thanks again, Gus. That was great.
Oh yeah, thanks. I'm glad it went okay. I was a little
Yeah,
nervous.
super.
Oh, didn't show at all.
Super great
Seriously,
job.
really cool stuff.
Yeah, yeah.
I just,
Really
can I ask you a quick question? Just if
Yeah.
you got a second?
Yeah.
So I didn't, have a chance to go and find my old botany textbook that has the life histories of these things. So when they infect the diatom, are they a diploid organism or are they haploid?
I don't know.
Okay,
I
so
mean-
I gotta look this up.
Again, I think I mentioned it. There is that sexual reproductive stage.
Okay,
I've really focused pretty heavily on them in relation to diatoms, so I haven't looked too much into the chytrid as its own life
Okay.
cycle. My co-author on that and my PI for the lab, Dr. Quant, doesn't look a ton into chytrids either.
okay,
I'm sure she would know, but
okay.
off my head,
It made me want to, go and see a life history and, figure out where everything fits together in these things. But what a cool thing. I mean, I see these things all the time in collections, and
I think that's what Tom said too, is he's
Yeah.
seen something like them or something similar all the time.
just,
But one of the broad things is it seems like since Dr. Cantor stopped doing this in past, nobody really has that really fine morphological knowledge to describe new ones.
yeah.
I just know
And really, and persistence to keep at looking at them beyond just incidental.
Mm-hmm.
Yeah, I meant to. I'm trying to look for a paper. I'll keep looking, Gus. There was somebody who studied the crash dynamics in Colorado, and it was like in the 70s or so. Just after winter, and it was on the western slope, like somewhere around Grand Lake, and I cited it in long ago, but I'll see if I can locate that again and pass it on. I thought it was really interesting because they showed those crash dynamics of Chytrids and Asterinella, and it was really interesting how you showed that temperature.
Yeah,
that was really neat.
I know we missed. I think the review was pretty thorough, but I do know we missed some stuff, especially some of the more recent work, because we had an early 2023 cutoff for. We can't just keep looking forever, and then so some of the things that have been cited too heavily yet or had just been published, I know we missed.
Yeah, cool. And it sounds like you're continuing to work on these.
Yeah, trying to
in culture.
That's awesome.
Encountering some small difficulties, but I think things we can get past. I think somebody mentioned TEM might be a better way to look at them. I think that's what I'm most interested in, is trying to get an infected diatom under a scope to try and really see if we can hammer out if it's that first method where it exploits some weakness in the frustule, or if it is mechanically creating an opening. Because I don't know, it seems it's hard for me to understand how it could drill a hole through the frustule, but it'd be really cool if it can.
I would think you'd be able to, I don't know if you've got sort of a model lake system in Colorado that you can hit regularly that has this issue, but I would think your old friend Asterinella would be an easy bet under the SEM, where you would get a collection, preserve it, probably with like a glutaraldehyde, a good fast, preservative and then go through this process called critical point drying.
Mhm.
And that would, not destroy the frustral and keep everyone together and happy. There is a I don't know if you've ever heard of critical point drying, but it was
I think I've heard it mentioned. The only way
yeah, it's a
crazy.
crazy, I don't know if anyone's got set up for it, but one thing
Yeah,
that we
Mark, now you can put
liquid.
wet material in and
Oh, sure.
avoid the artifacts even of that kind of drying, like using a low kV.
Yeah, but
I've
you may not,
taken wet material and
Can
put
you-
it in the SEM.
can you sputter coat
Is
or
that
not?
at CU?
You don't need to.
Oh, wow.
CU probably has it. I've done it at the USGS lab. But it's you use a very low kV, and so you can put in wet. We did that with Didymosunia stalks. Just take a rock and you can see things while they're still attached, and they don't have the. They still, even with critical point drawing, there's still some
Artifacts.
messed upness, that artifacts.
Yeah.
Yeah, really good.
Sarah, when you did that Didymo stuff, did you fix it first?
Mm-mm.
You didn't. You just put in live stuff. Dirty dogs. Wow. That's crazy.
Yeah, it's an amazing thing that SEMs can do now with,
For sure.
yeah, we drove down to Denver and picked up some cobbles out of the creek on the way there.
I love it. I love it. Well, sounds good.
Well, thank you again for
yeah.
you, me, and including me in this.
Yeah, great to see you, Gus. Thanks so much.
Thanks. Great talk. Yeah, appreciate it. All right. Good to see everyone.
Good to see you. Enjoy the.
Okay, bye.
Bye.
Take care.